right, well, you guys have made it. It's the last day. Welcome back to Bio 1120. I've been having a good time talking with you guys this last semester, and it's a bittersweet kind of thing for me. I'm a little sad that we won't be having this party anymore, but I'm also happy that we'll be moving on to some other things. Here's the deal, right? I told you about this on Tuesday. It's in the syllabus. There is an optional review session that you can come to on Monday to help you get ready for the final if you like. In my mind, this optional review session is essentially just office hours for me, but in a bigger room, right? So if you've come to visit me during my office hours at some point during the course of the semester, expect the same sort of vibe. It's just there'll be more people hanging out and eavesdropping on what it is that you and I are talking about. Again, anything you ask, I'll be happy to give you my best answer to. I'd like to go from about 10:30 to about 11:45 on Monday morning, and it's going to be in 116 Health Sciences. And then the big finish is the final exam for the class. One more time, if you hadn't heard, there's going to be a final exam. A couple people have gotten the idea that the final exam for this class is on Thursday of next week. Let me tell you categorically, it is on Tuesday of next week, right? And this slide sort of says it all. You're going to have 58 multiple choice questions. Come with a pencil so you can fill in the bubbles on one of those Scantron forms again. 58 multiple choice questions. For those of you who are in section 90, we're going to ask you to go to 101 Fawcett at 8 o'clock so that you can take the test there. There won't be enough room for you in 120 Med Sciences with everybody else. But for those of you who are not in section 90, excuse me, come to 120 Med Sciences. That's the room that we're in right now. And we'll have the exam ready for you. Starts at 8, ends at 10. I'm thinking 45, 50 minutes should be plenty for most of you to take the tests. It's not, you don't need to pack a lunch or have a snack break in the middle of this test. If you're looking for a good guide as to what the test is going to look like, one more time, I think this will be the last time you hear me say it, look at the old exams on the pilot page of the course, right? What I did is I started with last year's test, I copied it over, and then I made some changes to it. And I made a fair number of changes to the test, but a lot of the changes that I made were involved my going back to previous versions of the test and pulling off some old favorite questions and moving them on to last year's test. All right? So if you want a sense for how cumulative it is, if you want a sense for how I phrase questions, what sort of things I'm thinking are important and asking about, look at the old tests that you can find on Pilot. All right. Oh, hey, just crossed my mind. I've been looking at the long-range weather forecasts, right? Today's the last day for the foreseeable future, like the next 10 days out, that the temperatures are going to break freezing, right? So dress warmly. You know, your mothers are telling you wear coats. What? Wear coats. Do that. All the right things that way. One of the long-range weather forecasts is predicting a wicked ice storm on Tuesday morning. Right? They're talking about ½ an inch to an inch of ice accumulation. I don't know what to tell you if there's problems on the day of the final exam. I've never had that problem in 15-some years. Do whatever you can to, if Wright State does not cancel classes, do whatever you can to fight your way in, because I don't know how to give a makeup exam for a final exam. There's really no provision in the syllabus for it. I think what might end up having to happen is that we'll just have you take a final exam sometime in January, and you'll have an incomplete hanging over your head for a couple of days. So try as best as you can to get here on Tuesday morning. One of the long-range weather forecasts looks a little scary. It's still pretty far out. There are others that say it's just going to be a nasty, rainy morning. So enough said. But, you know, pay attention to the radio and such. If Wright State cancels classes, I'm sure there will be something that's also said about a day to come in to take a makeup exam. Right? They'll reschedule. So they'll say something like, if you had classes or final exams scheduled for Tuesday, come in on Saturday or whatever. Something like that. Okay, so enough about the doom and gloom of a final. Let's talk about the last part of this class, right? So I left you hanging in lecture on Tuesday talking about DNA sequencing. And it's not such a bad thing, because a lot of what I'm going to talk with you about today with genomics is I'm going to talk with you about what comes as a result of actually doing sequencing. And better, how it is you go about sequencing a really big genome like yours and mine. 3 billion nucleotides. It's no mean feat to be able to determine the order of each of those G's, A's, T's, and C's. But again, I left you just saying a few words about how sequencing is done, gave you a sense, I hope, of how it is that it's changed over the years. In the beginning, in 1976, Maxim and Gilbert got a Nobel Prize for figuring out a way to determine nucleotide sequences using a fairly brutish, bust-them-up kind of approach to breaking up DNA molecules that let them, again, ultimately determine the order of nucleotides in a particular part of a genome. And the essence of what they did is this, is they took a fragment of DNA. I told you on Tuesday that fragment of DNA was a clone. It came from the human beta globin gene. And what they did is they took that fragment of DNA, they had a bunch of this DNA, almost to the point where you could see it and hold it in your hands, and they radioactively labeled both ends of it. Okay? So they put a little glowing beacon, radioactive phosphorus, P32 is exactly what they used, but they put a little glowing radioactive beacon on both ends of the molecule. They used a restriction enzyme to get rid of one of the ends, and they could tell from size fractionation, you know, this is the smaller piece, we're not interested. And they were left with a stretch of DNA that had a radioactive label on one end. Okay? And then what they did is, Wally Gilbert's background is he's an organic chemist, and he worked up a cocktail of nasty chemicals that, when it's exposed to DNA, will bust up the DNA molecules a lot of the time, not always, but a lot of the time when there's a guanine within that DNA molecule. And he had another nasty cocktail that would bust up DNA molecules, but only when there were purines there, an A or a G. And another nasty cocktail that busted up DNA molecules, but only, most of the time, and only when there was a pyrimidine there, a T or a C. And one last cocktail that was really selective and only busted up DNA molecules where you found a C. Not a T or a C, just a C. And what ends up happening is, when you treat a DNA molecule that's got this glowing radioactive phosphorus at one end, and a string of nucleotides here, when you treat this molecule in this test tube, excuse me, you will generate a set of fragments of DNA molecules that have a glowing radioactive phosphorus at one end, but differ in length from one another, because sometimes, say this spot here is a G, sometimes you'll break the DNA molecule here, and that'll give you a really short fragment in this test tube. It's going to have the glowing phosphorus and one nucleotide and nothing else. But sometimes it won't break the molecule at this G. Instead, it'll break the molecule at, let's say this is a G, and now you've got a much bigger fragment, one that's 1, 2, 3, 4, 5, 6, 7 nucleotides long. And so in this tube, you're going to have a bunch of fragments of DNA. The fragments are going to differ in length, and the length will be dependent upon where along the length of the original molecule there was a G. Got it? Pretty simple, pretty straightforward, right? In another tube, you can do the same thing that generates a set of fragments that show up that correspond in length to where C's were located. And when you've done this for all 4 of these tubes, what you can do is run a gel, put all 4 of those fragments in a separate lane on your gel, strictly speaking, these were acrylamide gels. They're close cousins of the agarose gels that you guys have used in lab. But you run a gel, you put your fragments on the wells of this gel, you run it and you size fractionate, you separate on the basis of size. And in each lane, you see a series of bands, and the bands will differ from one lane to the next. But what you can do is pretty easily read up those bands and say, hey, there must have been a G at the beginning of the molecule. Oh, and the next largest fragment corresponds to where you would break if you had a G and an A, but you didn't break where you had a G, therefore it must be an A. You get what's called a sequencing ladder on this gel. You literally can climb up the ladder, walk your way up the ladder, nucleotide by nucleotide, and determine the sequence of the nucleotides in this original fragment of DNA. Again, lots of really toxic, nasty chemicals to pull this off. For goodness sakes, it busts up DNA. For goodness sakes, you use DNA. You like your DNA. You don't want it to get busted up. It wasn't elegant. It wasn't pretty. It wasn't even safe, but it got the job done, and it got Maxim and Gilbert a Nobel Prize. It wasn't long, though. It was just a few years, about 5 years, before a safer and better alternative to the Maxim and Gilbert approach swept through molecular biology laboratories, the Sanger approach. It's called also the dideoxynucleotide sequencing method. The difference, the principal difference, Maxim and Gilbert's approach is chemically based, right? The Sanger approach is enzyme-based. And what Sanger did is say, hey, let's tap in to some of the neat molecules, specifically the DNA polymerases, that living things use to access and make copies of that information in DNA molecules. And so Sanger's approach, instead of using chemicals, is really much more focused on some enzymes, and specifically DNA polymerase. And Sanger comes into this knowing what you know about the idiosyncrasies of DNA polymerases. Two things, right? They sit down and add nucleotides on only to existing double-stranded regions, right? And the other idiosyncrasy is they only go in a 5' to 3' direction, never 3' to 5'. So DNA polymerases all have those two rules. And knowing those idiosyncrasies of DNA polymerase, excuse me, Sanger knew that if you had primers, just like the primers that we talked about on Tuesday for PCR, if you had primers and you let them stick to complementary sequences on a template DNA molecule, you could make a sweet spot that a DNA polymerase might want to sit down on. And I tell you, as I'm looking at it, I see this primer looks like it would base pair really nicely with the beginning of this molecule down here. So you let this primer anneal, hybridize, base pair, all equivalent terms, with this template. DNA polymerase will think that's a nice place to get started, and it will add nucleotides to the primer in the 5' to 3' direction. So here's the 5' end of the primer, here's the 3' end. It's going to start adding nucleotides to the 3' end, and it's going to use the template molecule for the information to extend that primer. Where you get the fragments of different lengths that were so helpful to Maxim and Gilbert is that in addition to the primer and the template in the DNA polymerase, Sanger is playing a trick on the DNA polymerases. And specifically, the trick is that a lot of the nucleotides that you feed to the DNA polymerase are just what the DNA polymerase wants: GTP, ATP, CTP, and TTP, adenine triphosphate, and the rest. Right? That's what DNA polymerase uses. That's what it's usually in the business of incorporating and using to extend chains when it's doing DNA replication. But the trick that Sanger played was this: in addition to those 4 nucleotide triphosphates, Sanger also added some dideoxynucleotides. I think the next slide lets me show you the difference between deoxy and dideoxy. Oh, it's coming, or it's not. Maybe not. Oh, it's this slide. How about that? I'm getting ahead of myself here. So deoxyribonucleotides are what DNA polymerases are in the business of using. Dideoxyribonucleotides do a number on DNA polymerase. Remember the difference between DNA and RNA? Deoxyribonucleic acid as opposed to ribonucleic acid. The deoxy in DNA is a reference to the fact that you don't have a hydroxyl group on that 2' carbon. Right? If this was ribonucleic acid, there'd be an OH right where the cursor is pointing. But this is DNA. It's deoxyribose, so that OH is missing. For a dideoxynucleotide, it's missing not just that 2' hydroxyl group, it's also missing the 3' hydroxyl group, dideoxy. DNA polymerase, when it bumps into a dideoxynucleotide, is thinking, oh, it's a nucleotide triphosphate, I can work with this. And it incorporates it into the growing DNA molecule that it's replicating. But the problem comes then from the fact that because that 3' hydroxyl is missing, it can't add any more nucleotides onto that chain. Cool, huh? It's called a chain termination event. The DNA molecule that's being replicated has a length that's fixed, and it's determined by what dideoxynucleotide got incorporated at each step along the way. If you put a fluorescent tag on each of the 4 different dideoxynucleotides, what you can get is a set of fragments. Here, the template caused it to sometimes stop at the C, at the G, which would correspond to a C here on the copy. Excuse me, sometimes it stopped at the next nucleotide. If it did, it incorporated a good C here, not a dideoxy one. And so this fragment is 1 nucleotide longer than this fragment. Sometimes it stops at this position, which means that it incorporated a good C, a good G, but it brought in a dideoxy A, and it can't go any further. You get fragments whose lengths are determined by the sequence of the template DNA and whose fluorescent color, what color this molecule will glow, is determined by which dideoxynucleotide was the chain terminating dideoxynucleotide. You use a different color for each of the 4 different nucleotides. And when you separate out these fragments then on the basis of their size and run them here through a capillary tube, you get a parade, first a red blip, then a green blip, then a purplish blip, another purplish blip, a greenish blip. And the output that molecular biologists will look at often looks a lot like this. These blips, right? The detector is seeing different flashes of light, and those get memorialized in these graphs. And depending on the color that you see, that corresponds to a different nucleotide. And you can read off the nucleotides as they're coming through that capillary tube. You can determine the order of the nucleotides on that template strand. With Maxim and Gilbert's approach, it was all they could do to get 50 nucleotides of sequence. With the Sanger approach, even in the old days when I was a graduate student, you could get 400, 500, 600 nucleotides of sequence per run. So right out of the gate, it's an improvement. It's also an improvement because we're working here with chemicals that aren't that bad for you, right? And so it's a safer procedure. You're getting better results, longer reads off of the DNA. By the time that I was out of graduate school and my wife was finishing up her PhD, the whole process became heavily automated. You just fed it to a machine, and the machines could easily, and still today, easily get thousands of nucleotides of sequence and do it with very little human involvement. And essentially now, a molecular biologist will give the DNA to a machine, some prep that's involved, and the machine will tell them, here's the 2,000 nucleotide sequence that came from the spot where you had me start to sequence. All right, so it's become a very automated process. And it sets us up then to talk about what you do with all that sequence information, and also a little bit about how you go about sequencing a really big genome. The first genomes that got sequenced, and let me take a step back, I guess. Have I even said what a genome is? I don't think I have for this class. I've thrown the word around a couple of times. Today's the day I need to tell you what a genome is. You know what? There's a simple definition. A genome is the sum total of an organism's heritable information, right? It's the total of the information that you could pass on to another generation, right? That's what a genome is. When we talk about genomes, if we're being really casual, that usually means the DNA sequence of the organism's chromosomes, right? Because that's where we tend to think, at least when we're thinking casually, that's where we tend to think all of that heritable information is stored. But as something of a little asterisk, maybe a little bit of an aside, you guys know, maybe it's not front and foremost in your thinking, but you know there's more to the information that gets passed from one generation to the next than just the nucleotide sequence. Remember I talked with you a little bit about imprinting, chromatin structure sorts of things. The way that DNA gets modified, methylated, acetylated, the way the histones get modified, phosphorylated, and acetylated, that information is information, and it does get passed on to subsequent generations. And also, things like chloroplast chromosomes and mitochondria chromosomes. There's information there too, and so don't downplay that. There's other stuff that gets passed from one generation to another. The phospholipid bilayers, for instance. You know how you make a phospholipid bilayer? You extend an existing phospholipid bilayer. There's no gene that codes for phospholipid bilayers, so there's structural stuff associated with cells that in a very broad sense you could consider also being part of a genome. Right? So a genome is the sum total of all that heritable information. For most purposes, we focus on the stuff that we've got the best handle on, and that is the nucleotide sequence of our chromosomes. So for the rest of what I'll be doing with you here today, that's what I'll be talking about. But, you know, bear in mind, strictly speaking, a genome is all the rest of that stuff as well. But the first genomes that got sequenced were the smallest ones, viral genomes. It was a big deal back in the late 80s when bacteriophage lambda's genome got sequenced. Bacteriophage lambda, we've talked about lambda, it infects E. coli. It's the thing that's looking for the lac permease protein on the surface of E. coli. Its genome's about 40,000 nucleotides long. Back in the days when you could get a couple hundred nucleotides of sequence for each reaction that you set up, sequencing 40,000 nucleotides was a pretty big deal, right? That's the sort of thing that would take a graduate student probably 3-4 years to pull off. So that was a big deal. And it was also a big deal, not just because of the work that was involved, but for the first time with these viral genomes that had been sequenced, you could see the blueprint that an organism uses. I mean, that's the story. That's how lambda pulls it off. There's no, oh, there must be some other protein that solves this problem. No, you've got the complete sequence of the genome. Whatever lambda's doing, it's got to make do with the information in that genome. And so those genomes got a lot of attention. And it got people really chomping at the bit to crack a more complicated organism than a virus, because the things that you could see lambda was doing boggled the mind. How it is that lambda was able to milk information out of all sorts of strange little tricks that it was doing. Boy, if lambda is that sophisticated, it's just a virus, what will E. coli do? And so it was a big deal when E. coli's genome got sequenced. E. coli's genome is about 4 million nucleotides long. When it got sequenced, It got sequenced by folks who could sequence 1,000 or so nucleotides at a time. Again, 3-4 years of graduate student lives were expended to determine the sequence of E. coli's genome. But the funny thing is when E. coli's genome got sequenced, you guys know how much E. coli gets kicked around in the lab, right? How often has it come up in the context of this course? Everybody assumed that pretty much whatever there was to know about E. coli, we already knew it, right? It's been beaten up in the lab for decades at this point. What are you going to learn from looking at its chromosomes? Funny thing, from looking at E. coli's chromosomes, it turns out only about half of E. coli's genes had been characterized in the decades of experiments that had been done on E. coli. So people were like, whoa, E. coli can make twice the number of proteins and could do like twice the amount of stuff that we ever even guessed based on decades of research. And so sequencing E. coli made people think, wow, there's cool stuff going on here. We should sequence other things. Wouldn't it be cool if you could sequence a eukaryotic genome, right? Ratcheting up we're raising the bar here in a big way. E. coli's genome is 4 million nucleotides long. The simplest eukaryotic genome is about 1 billion nucleotides long. Yeast has a genome that's about 1 billion nucleotides. So it's twice as much work. If it took 3-4 years for a graduate student to sequence E. coli, at a minimum, it's going to take twice as long to sequence the yeast genome. But you put a team of people on it, the methodologies improve a little bit, you get the sequence of the yeast genome. Yeast has been beaten up almost as much as E. coli within laboratories. And it turns out that less than 1/3 of yeast's genes had been characterized before the sequencing of the yeast genome. And people were saying, no way, how can this be? I've been experimenting with yeast for decades. I had no idea it had this number of genes. And so they started to take a look at what do these genes do? And one of the nice things about working with E. coli and with yeast is you can reach in as a scientist and you can mess up stuff and nobody bats an eye, right? You can reach in and you can obliterate the largest gene on the largest chromosome in yeast and see what this mutant yeast does or how it's different than a yeast cell that you hadn't done that to, right? People did that. In fact, that was the first experiment that folks did. With the yeast genome sequence in hand, they said, biggest chromosome with its biggest gene. Nobody knew that this gene existed before we had sequenced the yeast genome. Let's see what yeast can't do if it doesn't have that gene. People thought they knew what yeast could do, the whole thing. Turns out that that one gene, the biggest gene on the first chromosome, gave yeast, as best as we can tell, the ability to do well, to be able to continue to live at low pH when a lot of acetic acid was present. Low pH without acetic acid, not a big deal. Acetic acid without low pH, not a big deal. Low pH and acetic acid, that gene seems to give yeast cells something of an advantage. People were saying, I didn't even know yeast could grow in acetic acid. I didn't even know yeast could grow at low pH. I didn't even know that there was a special issue. People discovered things about yeast from having the genome in hand and then being able to do some experiments with that information. So people are getting excited. And this stuff all then lays the foundation for what has been called the molecular biology moonshot, right? By the end of this decade, we will land a man on the moon and return him safely to Earth. John F. Kennedy said that and set off the whole space race. Well, it's part of the space race, and it gets a man on the moon. Cool thing about the moonshot is that at the time that Kennedy said, we're going to put a man on the moon and return him safely to Earth, everybody, Kennedy included, said, we can't do that right now, right? There's going to have to be some discoveries some breakthroughs that happen. Maybe if we put our mind to it and throw resources, money at it, we'll solve the problems and we'll make it possible. Because right now, I don't know how you're going to pull that off, but by golly, we're going to give it a shot. And along the way, as you're solving those problems, to pull off this feat that you couldn't do when you first said you were going to do it, lots of other cool spin-offs, discoveries happen along the way. We can thank the moonshot, the Apollo program in the 60s, for Tang. Any of you have Tang this morning with your breakfast? The powdered orange breakfast drink. It's kind of gross. People don't drink it that much anymore. It's still available, right? One more product placement for you in this class. Maybe I can slip in a few more today. But Tang came from the moonshot. Nobody had ever thought of dehydrating orange juice before that. Velcro. I bet half of you in the room have something with Velcro on it today. Velcro came from the moonshot. Nobody ever had even a vague idea about Velcro until they started playing around with how we're going to get a guy to the moon. Computer chips taking the place of vacuum tubes, those sorts of transistor sorts of things, all from the moonshot. So stuff gets figured out along the way from the investment to solve these other problems, and you get these cool sort of surprise discoveries as you're doing the work. So sequencing the human genome in the late 90s was considered to be the equivalent of the molecular biology moonshot. Folks made a proposition to the federal government in the United States and said, hey, think of all the cool stuff that we'll discover if we can sequence the whole human genome. Look what we found from looking at Lambda, E. coli, from yeast. We should find some other cool stuff. We'll find that human beings have capabilities and make proteins that do stuff that we had no clue that we could do. We need money to solve these problems. Give us $4 billion, $1 for each nucleotide in the genome is the way it worked out. Give us $4 billion, and in 10 years, we'll deliver to you the sequence of the human genome, even though right now we don't know quite how it is we'd pull it off. You give us that money, we'll figure out how to get past these stumbling blocks, we'll discover some cool stuff along the way, and we'll give you the human genome at the end. And the federal government, you gotta love them, right? They bought it, right? And they gave these researchers the billions of dollars that they asked for, and they turned them loose, and off they went to sequence the human genome. They developed a 4-part strategy to sequence the human genome, and that strategy is still the strategy that we use now 20 years later whenever you want to sequence some other complicated organism's genome. And the 4 parts of that strategy are spelled out here as the first 4 bullets, the first 4 lines, essentially, of this lecture outline. The first step in the process is establish a genetic map, a linkage map, a recombination map, if you prefer. That's how I've referred to it a couple of times in this class. Then you need a physical map. You need to do the actual sequencing. And when all of that is done, to make sense of things, you need to do that 4th step, the comparative genome analysis part. So that's the 4-step process for sequencing a genome. Let me say a few more words to you about each of those steps. Okay, so humans have 22 autosomes and 2 sex chromosomes, right? Total of 23 chromosomes and pairs of chromosomes in all of our cells. A good place to start is, look, we know you got these chromosomes. You guys, some of you were lucky in lab with the polyheme chromosomes and Drosophila, right? You know you can see bands on these chromosomes. And cytogeneticists for years had been able to figure out with recombination mapping experiments where along these chromosomes in their bands certain genes resided. Right? You know how to do that with recombination mapping for fruit flies. The same sorts of tricks can be done with human beings. It's just you have to wait for those mating experiments to happen of their own accord as opposed to setting them up as you would with fruit flies. There's also this trick called fluorescent in situ hybridization, FISH for short. And what happens with FISH is you can take a cDNA for a gene. I talked to you about cDNAs on Tuesday. It's essentially a messenger RNA that has been reverse transcribed, right? You can take a cDNA, label it with something that fluoresces, the same sort of dyes that got used for DNA sequencing in the automated Sanger method from a few minutes ago. You can take your fluorescently glowing cDNA, wash it over a set of chromosomes with their banding patterns, and see what parts of the chromosome light up with that cDNA. When you see what part of the chromosome lights up with the cDNA, guess what? That's where that gene hangs out. If it lights up a little bit of chromosome 1, I know where that gene is. It's somewhere on chromosome 1. If it lights up chromosome 16, it's on chromosome 16. And you can use those sorts of FISH approaches in conjunction with more conventional mating experiment, sort of classical Mendelian genetics, to figure out the lay of the land on the chromosomes. You know what genes are on which chromosomes, and you know their relative order. Not a bad place to start if your ultimate goal is to determine the sequence of that genome. Good to know where the points of interest are, right? So genetic mapping, linkage mapping, augmented with FISH gives you a good sense of the lay of the land. Then you can take those chromosomes, and you can use tricks like flow cytometry to get a pool that has just chromosome 1 in it, or just chromosome 2, or just chromosome 3. You can separate out the chromosomes, and then you bust them up. All right? You bust up chromosome 1 into a whole bunch of different fragments. Right? Random sort of buckshot, it's called shotgun sequencing. You bust it up into random fragments. You clone those fragments, and you make a library. This is a chromosome 1 specific library. But you make a library, and what you can do is use your sequencing tricks to determine the first 1,000 or so nucleotides at this end of this clone, and the first 1,000 or so nucleotides at this end of this clone. You can make probes based on that information that you can wash across your library of other chromosome 1 fragments and see which ones light up. And what happens then is you start to get overlapping clones. All right? See how that can happen? The last 1,000 nucleotides of this clone matches the first 1,000 or so nucleotides of this clone. And the last 1,000 or so nucleotides of this clone can be found at the beginning of this clone. And you can get then a set of ordered clones. You can have in your hand clones that are fragments of chromosome 1 that let you walk from the beginning of the chromosome to the end of the chromosome. You want a gene that's close to one end of chromosome 1? I got a clone for that. You want a gene or a clone that's from the middle of chromosome 1? I got a clone for that, right? And you can march your way across. And once you've got this physical map with these overlapping fragments, these overlapping clones, you can do the next step in the process, which is roll up your sleeves and sequence each of those fragments. You don't need to sequence every clone. You just need to sequence a set of clones that lets you walk from the beginning of the chromosome to the end of the chromosome. All right? And it's not too hard at that point to piece everything together, because this chunk of sequence, you know, comes from this clone, and this clone comes after this clone, which comes after this clone, which comes after this clone, and so forth. So you can piece the whole thing together. So those three things get done for the human genome, and they're still done today for any other genome that somebody wants to sequence. You want to sequence the maize genome? Don't bother, corn. Somebody's already done it, and they used this strategy. You want to sequence the chimpanzee genome? Don't bother. It's already been done. This is the strategy that was used to do it. There are over 1,000 eukaryotic genomes that have been sequenced now, right? And people are still chugging away sequencing other eukaryotic genomes. All the time, they use that three-pronged strategy. And at the end, for the human genome, you've got 3.2 billion G's, A's, T's, and C's in a particular order. When the human genome was being sequenced, there were actually two groups that were competing to sequence the genome. There was the sort of federally funded group using the $4 billion from the federal government. And then there was a private group that was doing it using their own money to a large extent. The competition seems to have borne some fruit. I said that they asked for 10 years to sequence the human genome. They came in at budget, but in just 6 or 7 years as opposed to the 10 years for sequencing the whole genome. So they were able to solve the problems and sequence the genome 3 or 4 years faster than they had said that it would take. The private group did pretty well, largely because the public group was making all of their information available free of charge. Right? So the private folks had some stuff that they knew that they weren't sharing. The public people were letting everybody see their strategies, their clones, their sequences. Right? But the private group claims that they finished the job before the public group did. And that's important to them because they then tried to patent the sequence of the human genome. Right? They filed a patent application. It's interesting on a couple of levels. At one level, let me impress upon you what an enormous amount of information 3.2 billion nucleotides is. It took 25 CDs to contain the information that was submitted as part of that patent application. 25 CDs. You guys, you know, that's a ridiculous amount of information. That's like, you know, like the New York City phone book 5,000 times. Right? It's just an insane amount of information. And so this group tried to patent it, and their patent application had 25 CDs to spell out the nucleotide sequence that they said was the human genome. Their patent application ultimately got rejected because it's hard for somebody to say just because they know the sequence that they own the sequence, because you own your DNA sequence as well, right? And their DNA sequence isn't that different from your DNA sequence. But they wanted to patent that information so that for the next 100 years, anybody who did anything with the information had to pay them bucks, right? The patent ultimately got rejected, but it was quite a fight. And it took a long while for that to get resolved. Nonetheless, lots of information. And part of the problem then with all of that information is how do you make sense of it, right? Because check it out. You can look at the human genome. It's available online. Go to GenBank, right? Just do a Google search for GenBank. Go to GenBank. You'll get a search field. What organism do you want to look at the sequence of? Type in Homo sapiens. You don't even have to. You could type in human or human genome, and it'll give you the whole human genome. Get ready. It's going to scroll through your screen for a long time. But it's just a bunch of G's, A's, T's, and C's. All right? There's nothing. There's no flashing light that says, and here's the insulin gene. Oh, and here's the beta globin gene. There's no annotations that come with a genome when you sequence it. What are you going to do with all of that information? That's where the fourth step in the process comes from. It's comparative genome analysis, and that's what this picture is supposed to make you start thinking about. How do you make sense of all of that information? The simplest way to make sense of it is comparative genome analysis. And the rationale goes like this. There are some differences between humans and chimps, right? At the DNA level, we're actually 98% identical. Between you and me, any two people in this room, we're 99.5% identical at the level of our DNA. In English, that means you have to look at 200 nucleotides a sequence before you find one difference between me and you. With chimps, you'll find two differences every 100 nucleotides. There's not that much difference in some sense between humans and chimps. But there are differences, right? Differences that give rise to the sorts of things you're seeing on the screen behind me. Those differences are probably pretty important, and we'd like to know what they are, right? The rationale for comparative genome analysis is this, is that if something is important to an organism, if there's a gene, if there's a stretch of nucleotides containing information, if there's something important to an organism, there's a really good chance that it's going to be conserved evolutionarily. It's not going to differ much from one individual in a population to the next. Mutations happen. Mutations happen all over our genome. But if a mutation happens to a really important gene, we don't see those individuals walking around, right? We've talked about the clinical term, right? They're dead. They're not here with us. We don't see them. There are some changes that happen to DNA that have consequences that are bad for the organism. We don't see those. The only things that we see changing are regions of the genome that, doesn't really matter, right? That a change happened there. No difference between the fitness of the organism if it had to change it or if it didn't have to change. When you look at pairs of individuals and you line their sequences up next to each other, there's a good chance that if you see differences between their DNA sequences, that's a part of their genome that doesn't matter that much. But if you see they're perfectly conserved, and not only perfectly conserved between pairs of people, but between people and chimpanzees, there might be something important going on there. Maybe there's a gene. Maybe that gene is coding for a protein that does an important job. And if you go further out, if you line up humans to dog DNA sequences, genomic sequences, and you see those tracts of DNA are the same, even though there's been 80 million years of independent accumulation of mutations, pretty good chance that's important. That must be a gene, all right? And if you go even further back and you find that there is commonality between a human DNA sequence and a yeast DNA sequence, 1 billion years of mutations, and you haven't changed those sequences, that must be really important, right? Because if you changed that any time in the span of the last 1 billion years, apparently all those organisms that had that change were dead. That's the rationale, and there's another payoff that comes from that rationale, because remember, we can do stuff to yeast that you can't do to humans. You can reach into yeast cells and you can obliterate a gene and see how messed up that baby yeast cell becomes after you've introduced that mutation. This looks like a gene that's common between humans and yeast. What are we going to do? Find some pregnant lady and ask her if she minds if we mutate one of her baby's genes? Or go to a lab and mess up the yeast equivalent gene and see how yeast is compromised compared to yeast that didn't have that mutation? Obviously, right? There's no Hitlers in the room, are there, right? Obviously, you go and you mess up the yeast cell. And if you find that yeast that has that mutation can't metabolize glucose anymore, guess what? I now know what that gene in humans is likely to do too. It's probably a gene involved in glucose metabolism. All right? And so what we learn from beating up yeast, both the old-fashioned way and this modern way of messing up genes deliberately, what we learn from studying yeast tells us a lot about the genes that we find in humans, what their function is. There are a lot of genes in the human genome that can't be messed up, right? Because you need those genes, right? The ones involved in basic metabolism, making ribosomes, metabolizing glucose, making guanine, making adenine, right? Those genes you can't find differences in, but we can recognize them because we found them in yeast. And so at the end of the day, this comparative genome analysis business is our single best bet at figuring out what the genes that we're finding in the human, where the genes are, what's a gene, and then what those genes are likely to do. But there's still some drawbacks, right, with this whole strategy, because again, don't forget there are differences between humans and chimpanzees, and we're not going to find, there's stuff that we can do that chimps can't do. We're not going to find the gene that lets us do that because it's apparently something different from what chimps can do. So we don't get the whole story, but we get a lot of it. So that's what's up with comparative genome analysis, and to some extent, comparative genome analysis never ends. And that's why there are still thousands of people who make their living sequencing eukaryotic genomes today. It's strategic. Sometimes you choose an organism because of its important commercial value. It's a crop, right? Something like that. Sometimes an organism gets chosen because it's responsible for a disease. Sometimes it gets chosen because it's going to give you a piece of a puzzle. Gee, you know, I saw this similarity between these two organisms. I want something sort of intermediate between them. Sometimes it's done strategically to give you those types of insights. And when all is said and done, here's a picture that shows you how you might line up those sequences. Look at this similarity here between a cow string of amino acids, this is the one-letter code for amino acids, and a mustard plant. This is probably Arabidopsis thaliana. This gets beat up a lot as a plant. Look at those similarities across, this is probably about 500 million years of evolution. There's a lot of these amino acids are lining up, and you can see these here in particular are absolutely conserved. Those must be really important to the function of that particular protein. Not only have we identified a gene, we've identified the really key amino acids. Those are probably part of the active site. They're involved in regulation. And so you get those types of insights from these sorts of sequence alignments. And if you've got an X-ray crystal structure for the mustard weed version of this protein, you can probably figure out by slipping in the cow version what the cow three-dimensional structure of the protein would look like as well. That's all bioinformatics. Another part of bioinformatics is looking at these relationships between genes. If you find there's a cluster of genes in yeast or proteins that physically interact with each other in yeast, they're probably doing some related function. And you can see here these genes are all related to each other. They're involved in secretion and vesicle transport, and they rarely interact with other things. You get this sort of map of interactions between genes. Again, all bioinformatics stuff, making sense of all that data. When you get all of that stuff put together, here's the big payoff, right? It's this newer idea, something that was only vaguely imagined in the 90s when people were working on sequencing the human genome. The big payoff that we can see right now is this idea of pharmacogenomics. In another way of describing it, personalized medicine. And so for pharmacogenomics, right now, today, it would cost on the order of $1,000 to sequence your genome. So the price has gone way down. It used to be $1 a nucleotide. Now for $1,000, not $3 billion, for $1,000, we could determine the sequence of every nucleotide within your DNA, within your genome. It's realistic to expect that in the next few years, it's probably going to get down to $200, $300. It's going to get to the point where an insurance company is going to say, hey, would you mind if we sequenced your genome? Because it would come in real handy for us as the insurance company to know if you are going to die young from cancer. Because if you will, we have a different insurance policy for you. It's the super high premium where you pay us a lot of money right now kind of approach, as opposed to, no, you look like you might be able to hang on for a long time. Got none of the indicators for cancer, none of the indicators for neurodegenerative diseases. We want you. Come get a policy with us, and we'll just take a little bit of money from you because you're going to be giving us a little bit of money for a long time. Right? So the insurance companies are going to be really interested in this approach. But you might be interested in this approach to some extent too. Let me give you one specific example. If you watch football games, if you read Sports Illustrated, these are things where I see. You ever see the ads for Plavix? You know what Plavix is all about? Plavix is a drug that costs about $10 a dose, right? $10 a pill is what it comes down to. And what you take Plavix for is to reduce the function of your platelets in your blood. It reduces your clotting time, right? And people get prescribed Plavix, particularly if they have a history of problems with blood clotting, and I'm talking specifically about heart attacks. And so if you've had a heart attack, there's a good chance that if, and not you guys haven't had heart attacks, but your parents, your uncles, your aunts, somebody you know has had a heart attack. A physician has suggested to that heart attack survivor, we're going to put you on a drug to reduce your blood clotting time. So that's going to, because it's been shown that you do that and you reduce the risk of future heart attacks and the severity of future heart attacks. Good. Who doesn't want that, right? You're at risk. You've demonstrated you're at risk. We get a drug that's going to reduce your risk in the future. Who wouldn't like that? For the small price of $10 a day, I'd pay $10 a day, right? Yeah, sign me up. If I was at risk and I thought that was going to help me, if it was going to give me an extra 10, 20 years of life, yeah. You can have that $10 a day for the rest of my life if it gives me an extra 10, 20 years. Of course I'll do that. But here's the thing. Plavix does the same trick, solves the same problem as a much cheaper medicine. It turns out that aspirin, and not even a whole aspirin, but a baby aspirin, does the same trick as Plavix does, with one little caveat, with one little caution. It turns out that within the human population, within the people in this room, probably about 10% of us are unresponsive to aspirin. Pretty much everybody responds to Plavix, but about 10% of us don't respond to aspirin. It doesn't affect their blood clotting time, all right? And so if you're a physician and you're worried about malpractice, or you really want to make sure this guy lives another 10, 20 years, what do? You should take a baby aspirin every day for the rest of your life, and there's a 10% chance that's not going to do you any good and you're going to die. Or take Plavix. I'm not paying the $10. I'm the physician. I'm prescribing it. You'll be fine. Your insurance will pay for it. The thing is there's a gene that we know of that you can look to within your genome that tells us if you are going to respond to the aspirin therapy or if you're one of the smaller fraction of people that needs the Plavix. All right? That's what pharmacogenomics is all about. Before you start taking Plavix, you should probably have that genetic test done. Look at your genome and see if you're one of the people who needs that special, expensive $10 a day drug, as opposed to the $0.25 baby aspirin dose each day. That's what pharmacogenomics is all about. You can see how that could extrapolate too, right? There's an enzyme, it's mentioned in your lecture notes, cytochrome P450, that detoxifies drugs and toxins in your liver. Some people have hyperactive versions of cytochrome P450. Some people have really slackered versions of cytochrome P450. Chemotherapy, you give somebody a drug to treat them for cancer, you got to play around as a physician for a number. It's a trial and error approach. Most people do pretty well with this concentration of this chemotherapeutic agent. But if you've got the super hyper version of cytochrome P450, we need to ratchet it up. You need to have more because your liver's taking it out of your system faster than we're putting it in. Or if you've got the really bad version of cytochrome P450, we want to back off a little bit because you're going to be accumulating the drug in your blood too fast if we don't take that into account. There's all sorts of stuff. Personalized medicine. You look at your DNA, you look at your genome, you look at your DNA, and physicians within just a few years are probably going to be able to say, you know what, you have this problem, and you would benefit from this choice of the 10 options that you have to deal with that problem. So that's what pharmacogenomics is all about. We wouldn't be talking about pharmacogenomics if not for all of this genome sequencing stuff going on. All right, now, I'll confess, the next 10 minutes, I'm going to go through things kind of quickly. I just want to toss out to you some cool things that also fall into this bin of stuff you learn from studying genomes. These are things that I just think are kind of cool. I'm hoping that they may have some relevance to you down the line. To some extent, I'm feeling like I'm indulging myself by telling you these things because, again, I think they're just cool. Here's this neat idea. It's called a C-value. C is an abbreviation for content, and that's sort of a shorthand way of talking about DNA content. Okay? Every organism, every species, let's put it that way, has a characteristic amount of DNA in its genome. E. coli's genome, all E. coli cells have the same amount of DNA. They have the same C-value. Human genome, all of us have the same C-value. Obviously, there are differences in our sequences, but the total amount of DNA is the same. And there's differences between species. And people, early on, like in the 60s, realized that you might be able to get smug when you looked at how much DNA you have as opposed to some other organism. All right? Coming into this, you should expect a virus would have puny, teeny, tiny little bits of DNA, right? It's just a virus. You can look down your nose at viruses. They've got very little DNA. That means they can't be very complicated organisms. You've got lots of DNA. You must be swell, right? That's the thinking here. E. coli, oh, a little bigger, more DNA than a virus has, but still much less than you do. Well, funny thing. There's this thing called the C-value paradox. That thinking bears out for the most part. The more DNA an organism has, the more stuff it can do, the more complicated it is, the more information it lugs around inside of all of its cells. Generally speaking, that's correct. But what makes it a paradox are the glaring exceptions to that general rule. All right? And so, have you ever wondered, with all the UFO sightings and such, Area 51 just down the road from us here, right? Have you ever wondered why the aliens haven't been spending a lot of time talking to people? What's with all the crop circles? Why are they spending all this time out in cornfields and wheat fields, right? Maybe what's happened, this is speculation, right? Maybe what's happened is aliens have surveyed the planet, and they've fallen prey to the C-value paradox. And they say, gee, look at this. This organism has lots of DNA. We should talk to them. Oh, but wait a minute here, Byborg, whatever the guy's name is. Wait a minute. This organism has even more DNA. It turns out that onions have about 100 times larger C-value than humans do. That sucks, right? Maybe the aliens are spending all this time trying to strike up a conversation, work out a trade deal with onions as opposed to people. Or corn plants, which also have larger genomes than we do. What's up with that, right? It doesn't seem right. And it gets tricky because, well, so check it out. So here's corn. Look at these here. Corn has a genome that's 2.3 billion base pairs long. Rice has a genome that's 0.4 billion nucleotides long. You guys know what corn plants look like. You guys know what rice plants look like. I don't see what is that, a 50-fold or is it a 500-fold level of difference in complexity between corn and rice. Something's not right there. All right, there's something not right about how big corn's genome is relative to yeast's. And look at this. These lily plants are 40 times larger genomes than ours. Again, that just doesn't seem right. And so that C-value paradox got people thinking, okay, it's a general rule, but what's corn doing with all that extra DNA compared to rice? And the answer that people kept coming back to, and it's still the best answer today, is a lot of what corn lugs around doesn't seem to be doing that much, right? Corn could probably have gotten by with a much smaller genome, 400 million nucleotides as opposed to 2,300 million nucleotides. And if that's the case, if there's a lot of extra stuff in corn's genome that it doesn't use, that it could be just as well off without, we got a word for stuff like that, right? There's stuff in your apartment. There's stuff in your car that you lug around with you that you're never going to use, the crumpled up gum wrappers, the empty water bottles that my daughters stockpile in the back seat of my car for no apparent reason. We got a word for that stuff, the stuff you lug around that's extra, that serves no purpose, that you would have been just as well enough off without. What's that word? You know it. It's junk, right? And that's the whole idea behind junk DNA. You've probably heard of junk DNA. The larger the organism, the more we start to think that a lot of the DNA that it lugs around with it is junk DNA. And that explains then this disparity, this sort of paradox between the size of a corn genome and a rice genome. And so in light of junk DNA, right, the next position that people adopted was essentially, okay, look, it's not so much how much DNA you have. It's not how big your C value is. It's how you use it, right? Size isn't what's important. It's what you're doing with all that information. What are you doing with that information? You got genes in there. The number of genes, that must be what we should be keying in on to help us feel good about ourselves, better than all the other organisms on the planet. And the number of genes also bears that same sort of trend. Look at this. This virus here, or this very simple bacteria, has only 1,700 genes. An interesting lesson to be learned from that, right? That might be like the bare number of proteins that an organism needs to be able to make in order to be free living. How about that? That's kind of a cool thing to have in the back of your head, because everything's got to make ribosomes, everything's got to metabolize glucose, everything's got to be able to replicate DNA, right? Citric acid cycle. All of those genes together, maybe that adds up to about 1,700. Maybe there's 100 extra here to make this thing special, but that might be the bare bones that you need to get by. But you get a little more complicated, like E. coli. That's got more than double the number of genes. It's got 4,400 genes. And as you go along here, let's find yeast. Yeah, look at that eukaryote, 6,300 genes. Of course, the simplest eukaryote would be head and shoulders better than the most complicated prokaryote. That seems to be the case. How about that? But check this out. Before you start feeling too smug about yourselves, at the time that this table was made, the thinking was there were in the ballpark of 21,000 human genes. Feels pretty good when you're comparing yourself to a yeast cell, but it kind of sucks to be you when you're comparing yourself to a rice cell in terms of the number of genes that it has. What's up with that? Rice has twice the number of genes as humans do? Doesn't seem right, doesn't seem fair. So, it's not how big your genome is that matters. Apparently, it's not even how many genes you have that matters. This is where we're at today. It's how many different proteins you make, all right? And humans are the all-time champ so far in terms of alternative splicing. We can make about 250,000 different proteins from our puny number of genes in our relatively small genome. That's the C-value paradox. And what's all that other stuff within the human genome? Well, in a word, a lot of it is junk. Less than 2% of our genome corresponds to exons, things that ultimately get translated into proteins. About 5% of our genome are introns. This is generous, but maybe 20% is involved with what's needed to turn genes on and off. And pretty much all the rest, there's a little bit, 15% that's just one-off stuff, but the majority of the DNA in our genome is highly repeated. It's the same sequences over and over and over again. Hundreds, thousands, literally millions of copies. And once again, I'll make a reference to a Simpsons episode. You guys have all been there. You've lived this. You've been assigned to write an essay for a class, 500-word essay, right? You're assigned to write an essay, a book report, right? 500 words. There's a certain expectation of information. Bart Simpson's approach to a 500-word book report, the book, 2 words, was 3, very. Let's go with very another, 496 times. Well, the book was very, 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 very boring. Done. Walk away. There's my book report. You know, how much extra information got conveyed by the 5th iteration of the word very as opposed to the 64th version of the word very, right? You got the idea across after the 2nd or 3rd time you said very. When you look at a genome like the human genome and you see a sequence that's 100 nucleotides long and yet repeated 1,000,000, I'm tempted to go blue, 1,000,000 frickin' times, right? That's junk. You didn't really need it 1,000,000 times. If you needed it at all, 2 or 3 would have probably gotten the job done just fine. So that's what's counting for a lot of our junk DNA. And so, what's junk, right? We get a lot of repeated DNA. Included within the repeated DNA are these neat things called gene families. Let me just take a few minutes to talk with you about gene families, and we'll be done. All right? So I keep coming back to the alpha and beta globin. They make the hemoglobin protein in your blood, partly because they've been studied a long time, and they're kind of foundational to our understanding then about a lot of other things, but also partly because there's a really neat story associated with the globin gene family. So there's 2 proteins. There's an alpha globin and a beta globin. You make 2 copies of each of the proteins, you push them together, and you got hemoglobin to carry oxygen in your blood. Through comparative genome analysis, right, we know something about where those globin genes have come from, how they've evolved over the course of about 1 billion years. Today, within humans, on human chromosome 16, that's where the alpha globin gene family resides. And on human chromosome 11, that's where the beta globin gene family resides. And here's what's up with these families. Alpha globin, within humans, most humans have 2 copies of the alpha globin gene. And they're right next to each other on human chromosome 6. This is the gene that gets transcribed and then ultimately translated to make the alpha globin protein. But as an embryo, you used a different version of the alpha globin gene. It's called the zeta globin gene. And here's part of the trick, right? Embryos. Picture this, right? Little developing baby deep inside of a woman, all right? The woman's got proteins that suck up oxygen, right? But the baby's buried deep inside of this woman, and it needs to suck oxygen away from the mother, right? And so it needs a globin that's got an even higher affinity for oxygen than the mother's is. Otherwise, it's not going to work too well for the baby. Zeta globin, the embryonic form of the alpha globin protein, lets developing embryos leach oxygen away from their generous mother's normal adult hemoglobin. So it was a good thing for some point in the past for another version of the alpha globin gene to show up that had a higher affinity for oxygen, but only got expressed early in development. That gene hangs out nearby the adult forms of the alpha globin gene. For the beta globin gene, there's the same story. There's an embryonic form, there's a fetal form, and then there are 2 copies of the adult beta globin gene. So where do these genes come from, these gene families? Well, about 500 million years ago, maybe actually 1 billion years ago, it looks as if there was just 1 globin gene, right? And then it got duplicated, And one of those duplicates accumulated changes that caused it to become the alpha globin that we know today. And the other duplicate accumulated the changes that caused it to become the beta globin we know today. At some subsequent point in time, the alpha globin duplicated, and you got an embryonic form. It accumulated some changes that caused it to get expressed early in embryonic development, but not late in life, and vice versa for the alpha globin. And the same thing's going on with the beta globin here. And what's happening is the way evolution seems to work. This has been described as evolution is a tinkerer. You need a gene with a new function. How do you get a gene with a new function? A protein that can do something different than proteins currently can do? There's two choices you get if you're Mr. Evolution. One choice is you just do random die rolls with nucleotides, and you hope that just by chance everything lines up just right. The minus 35, the minus 10, the plus 1, all the regulatory sequences, the open reading frame, translational start sites, splice junctions. It could happen. You roll the dice a couple quintillion times, and you might get a gene with a new function. Or you can take a gene with an existing function, make a copy of it, and then poke away at both of them. All right? If one of them gets a change that gives it a new ability, and that new ability comes in handy for the organism, keep it. It's under selective constraint. It's not going to change anymore. And you're one leg up on all the competition. A price you pay for that tinkering approach, if you want to duplicate genes and poke around at them, a price you pay has got to be the accumulation of what are known as pseudogenes, false genes. Genes that have accumulated a change that was just a really bad idea. What if we, instead of having AUG as the start codon, we have AUU as the start codon? When we change that G to a U, end of the road, that's a pseudogene, a derelict gene, and from that point on, it doesn't do anything useful, and it just keeps accumulating changes until eventually you can't recognize it as having once been a copy of an alpha globin gene. And so that's what gene families are all about, and how it is that we get genes with new functions. For the most part, is you take a gene with an existing function, you duplicate it, and then you tinker with it. And I tell you what, that's the important stuff. I told you the stuff that I know is going to be on the final. You'll see on Tuesday. Right? I wanted to make sure I talked with you about how genes with new functions come up. I feel like I've done my job. I'm looking forward to reading your student evaluations. Do those online for me, please. Tell me what you thought worked and what didn't work. Good luck with the final exam. I'll see some of you on Monday for the optional review session. And certainly if you have any questions or if I can help any other way, send me an email and I'll get back to you as fast as I can. Good talking with you guys. Good luck in finals. Have a good weekend.